Hello everyone. This class will be Introduction to Renal System. Renal system consists of pair of kidneys, ureters, and urinary bladder and urethra. Some of the functions of kidney are their excretory, some of them are the homeostatic functions uh, and endocrine functions. Excretory functions are formation of urine by three mechanisms, that is by glomerular filtration, tubular reabsorption, and tubular secretion. They also help in excretion of urea, uric acid, creatinine. Homeostatic functions like they're important for maintenance of constant internal environment by maintaining water and electrolyte balance through the buffering actions. Some of the endocrine functions of kidney are involved in renin secretion during renal ischemia and release of renal erythropoietic factor during hypoxia, release of local hormones like prostaglandins, kininogens to regulate mainly the renal blood flow. They're also important for formation of the vitamin D3. They're also important for regulation of normal arterial blood pressure. It is important in the hydroxylation of 25 hydroxycholecalciferol to 125 dihydrocholecalciferol, that is to activate to activated form of vitamin D. Nephron. Nephron is a functional unit of kidney. There are 1.3 millions of nephron per kidney. Each nephron is formed by two parts, that is renal corpuscle and renal tubules. Renal corpuscle is formed by two portions, glomerulus and Bowman's capsule. Glomerulus, the diameter is around 200 micrometers. Glomerulus is a tuft of capillaries enclosed by Bowman's capsule. Blood enters glomerulus through afferent arteriole and leaves through the efferent arteriole. Bowman's capsule is a dilated portion of a nephron. Renal tubules. There can be proximal convoluted tubule, that is PCT. Part the pars it has different parts: pars convoluta and pars recta. That is, pars convoluta is a convoluted portion, whereas pars recta is a straight portion. Its wall is made up of a single layer of cells whose luminal edges have striate brush border due to the presence of microvilli. Tubular cells are rich in mitochondria. The tubular cells are connected by tight junctions at their luminal edges. But there is a space between the cells along the rest of their lateral borders called lateral intercellular space. Loop of Henle. It consists of a descending and ascending limb. The descending portion of the loop and proximal portion of the ascending limb is made up of thin and permeable cells, whereas thick portion of ascending limb is made up of thick cells containing many mitochondria. Distal convoluted tubule, that is DCT, has few microalveoli present. There is no distinct brush border here. Collecting duct. The distal convoluted tubule opens into collecting duct. Epithelium made up of principal cells, that is P cells, and intercalated cells, that is I cells. P cells, they're mainly involved in sodium reabsorption and vasopressin stimulated water reabsorption, whereas I cells are concerned with acid excretion and bicarbonates. Based on the situation of renal corpuscle, nephrons are classified as a cortical and juxtamedullary nephrons. So this is the typical part of the nephron, which will be comprises of glomeruli and the tubules. Tubules, it can be proximal convoluted tubule, which is convoluted. Again, it has a straight part. Then we have a loop of Henle. Then distal convoluted tubule. Then we have collecting duct. And in the different parts, the organization of the cells, they are going to differ a bit. Where in proximal convoluted tubule, the cells have a microvilli and they possess brush borders. Ultrastructure of glomerular membrane. Ultrastructure of glomerular membrane refers to the membrane that separates the blood glomerular capillaries from glomerular filtrate in Bowman's capsule. It consists of three layers. Capillary endothelium. It is perforated with thousands of small spores called fenestrae. That is, pores have a diameter of around 70 to 90 nanometers. The endothelial cells have fixed negative charges that hinder the passage of plasma proteins, that is, which are negatively charged. Basement membrane consists of a meshwork of collagen and proteoglycan fibrillae that have a large spaces through which water and small solutes are permeable. They effectively prevent filtration of plasma proteins because of strong negative charges associated with proteoglycans. Layer of epithelial cells. Visceral epithelium of Bowman's capsule is constituting the epithelial layer. It is formed of special cells called podocytes. They have foot-like processes, that is, which are also called as pseudopodia, towards the basement membrane that encircle the outer surface of capillaries. Food processes are separated by gaps, uh, which are called as slit pores, having a diameter of around 25 nanometers, through which the glomerular filtrate moves. The epithelial cells, which also have a negative charges, provide additional restriction to the movement of plasma proteins, especially negatively charged one. So this is the diagram showing the glomerulocapillary epithelia, and you can see the three layers: endothelial cell, basement membrane, and the podocyte layer. We can see the podocytes, they're going to send processes which are going to encircle the capillaries, and they will form the glomerular filtrating membrane. Net diameter of pore in glomerular membrane is 8 nanometer. The pores allow passage of neutral substance up to 4 nanometer. Around 4 to 8 nanometer particles can pass based on the charge and size. For example, albumin, which is having a diameter of 6 nanometer, due to its negative charge, it is not filtered through the glomerular membrane. Particles greater than 8 nanometers are completely withheld. Negatively, the negativity is lost in some of the renal diseases. Then negatively charged substances like albumin is filtered. Hence, proteinuria occurs. Glomerular diseases like nephritic syndrome, the pore size increases. So here in this table, some of the differences between cortical nephron and juxtamedullary nephron, they're given. Cortical nephron, they're almost 85%, whereas the medullary nephrons, they're 15%. Cortical nephron occupies 2/3 of the cortex and very small portion of the medulla, whereas juxtamedullary occupies 1/3 of cortex and most other parts there in the medulla. Loop of Henle is short here, loop of Henle is long in medullary nephron. Descending limb of loop of Henle contains thin segment, whereas ascending limb contains thin and thick segment, whereas in juxtamedullary, both descending and ascending contains thin segment. Cortical nephron efferent arteriole constitute as a peritubular capillaries, continue as a peritubular capillaries, whereas efferent arteriole here continues as a vasa recta. The cortical nephron associated with the formation of urine, and medullary, it is associated with concentration of urine. The efferent arteriole is smaller than the afferent in cortical, whereas efferent arteriole has the same diameter as afferent in medullary nephron. So this is a structural difference where you can see the loop of Henle in the cortical nephron, they are short, and in the medullary nephrons they are very, very long. The cortical nephrons, they are associated with formation of the urine, whereas juxtamedullary nephron, they are associated with the urinary concentration. Thank you.